Anyway, thank you very much for having me here, and thank you, Karen, for the introductions and for inviting me to come tonight. So I'm very happy to be with you because learning about living soil has become a passion of mine. And tonight I'm hoping to give you just a glimpse into this incredibly exciting and intricate world that is right beneath your feet. And a lot of people don't even know that it all exists. Knowing how your plants interact with soil life is the beginning of knowing how to care for them in the way that nature intended. When you foster the life within this soil ecosystem, then your rose plants, which are part of that ecosystem, are healthier and become resistant to diseases and pests. On our journey today, we will talk about the basics of soil and how living soil contributes to healthy plants. But there is more to this story. But even though there is more, what I'm going to tell you tonight should be a good enough start for you to get going. I'm hoping you'll come away with understanding some of the complexities of what happened underground. And while you might think that this information pertains only to plants in the ground, it also pertains to plants in containers and pots, raised beds, etc. And I'll be showing you some pictures of life in the soil, real life ones, later today. Many times during this talk, I'm going to be saying the words soil microbes. When I'm saying that, all I'm talking about are the little critters that you can't see without the aid of magnification, such as bacteria and fungi. So here are the topics we're going to be covering this evening. We will begin by asking the question, well, what is a healthy living soil? We're going to look at roots, since that is the part of the plant that interacts with the soil. Then we will discuss where nutrients are located. And do you know already where those nutrients are located? Some of you do, I bet. We'll talk a bit about the soil food web. Now, can you raise your hand if the term soil food web is new to you? Okay, that's great, hands down. And how many of you are very familiar with it? Somewhat familiar with it? Okay, there we go. We will discuss ways that you as gardeners can unintentionally damage soil and ways that you can help support the living soil in your own gardens. Then we will look at some photos, as I just told you, of actual soil microbes that live around roses. I'd appreciate if you would save your questions until the end, unless you really need something clarified or you're just not going to understand anything. In that case, raise your hand and I'll take your question. Okay, so what is healthy living soil? You may have heard people refer to healthy soil as having good tilth, that it's loamy or that it's crumbly. And that is all true, but we're going to look at it in a little more detail. When you look at healthy soil, you should see a rich, dark color. Smell it, and it should have a good smell. It should never have a pungent odor. And it is full of life. But what really matters is how your soil functions. If you put water on top of soil, it should infiltrate or soak in reasonably quickly. Water should go deep into the soil profile and spread throughout it. And in healthy soil, water should be held so that it's available for later use for your plants, not just put it in, it drains through, and then you're done. Some of it's got to be held back. And roots can easily penetrate and grow as deep as they need to grow, or as deep as they're genetically programmed to do. And plant nutrients are available in small quantities all the time. So that's how a healthy living soil should function. When I talk about the soil being full of life, I am referring to the larger creatures as well as the smaller creatures that affect the soil environment. But tonight we will mainly focus on those smaller ones. Life in the soil also includes plant roots. They are key to the soil ecosystem. Soil life is crucial in creating soil structure that enables water to move into and around the soil and to hold on to the water as well. The dark color, the good smell, and the availability of nutrients can also be attributed to this life in the soil. So what does it mean for water to infiltrate into and percolate through soil? If you look at this image, water infiltrates into the soil right at the soil surface, and that is represented by the green on this slide. Once it infiltrates, it then percolates through the soil, represented by that brown area. And water eventually reaches the water table and then below it. So that's how it works, and that's seen in the lighter blue area. So what makes it possible for water to infiltrate into and percolate through soil? It is because a well-functioning soil has spaces between those particles, and those are known as pore spaces. Notice the image on the left. The brown represents soil particles or aggregates, and we will talk about that word aggregates shortly. The white represents those pore spaces. There are plenty of pore spaces around the soil particles to allow the water and air to flow, as well as let those roots grow deep and wide. And those pore spaces, remember, hold on to some of that water and prevents it from flowing through to the water table. So it is available to plants for later use. So now let's look at that image on the right. You notice that at the top, there are no open pore spaces, okay? So when it looks like that, you know you have a compacted soil. In this case, it is a surface compaction. Compaction can happen on the surface or happen farther down in your soil. In a compacted soil, water cannot move the way it should. If the top of the soil is compacted or crusted, well, then water cannot infiltrate into it, and it's going to run off and most likely take some soil with it. If there is a compaction layer deeper down, then water cannot percolate in and then plant roots cannot go as deep as they desire, because think about that solid piece being down below. They just stop there at the compaction layer. So now that you have an overall idea of how soil is supposed to function, we're going to go a little deeper. So this chart shows the 4 basic components of soil and their ideal percentages. On the left side of this diagram, you see air and water. This represents those pore spaces we were just talking about. Those pore spaces can and should make up about 50% of the volume of your soil. Think about how fluffy that is. And then whether it's air or water depends on what's happening. If there's a rain event or you've just used your sprinklers, run your irrigation, there's going to be a lot of water in there. And then as the water flows and percolates through down to the water table and all, then it draws in the air. So places where the water is not being held, then air can come. So that air and water space is constantly fluctuating. So you can think of this 50% as just being empty space. Now on the right, you see minerals, which in an ideal soil is about 45% of the soil volume. Minerals are simply the rocks that have been broken down into smaller and smaller particles until they become the sand, silt, and clay that you have in your home gardens. If you look at the images on the right, sand, which is shown in that big yellow circle, is the largest particle. If you have lots of sand in your soil, well then you also have large pore spaces. And if any of you have sandy soil, actually raise your hand if you have sandy soil. Yeah, you know that water drains super quickly through there. Okay? Clay is represented by that tiny little red dot in the diagram. And it is the smallest particle. Clay is great because it holds on to nutrients and it holds on to water very tightly. But unfortunately, if you have more clay in your soil, you'll also notice that it's pretty slow draining. The other particle is the silt particle, which is shown in blue. It's more of the medium-sized particle, and it has a little bit of the qualities of sand and clay. There are tests you can do to determine the percentages of sand, silt, and clay that you have, and determining those percentages will tell you what kind of soil texture you have. There is a link to a texture test by field in the resource section if you'd like to figure out your soil texture, and we'll be sending those out, but just so you know, it looks something like this. It's a nice flow chart where you can get soil in your hand and rub it and see how it feels and follow through the chart, and then you figure out what kind of texture you have in your soil. And on a side note, a loamy soil is one that has a fairly even mix of sand, silt, and clay. But if it's a sandy loam, it has just a little bit higher bit of sand, and a clay loam has a little bit more clay. Now, the smallest percent of soil is the organic matter, but it has the greatest impact on how your soil functions. The organic matter is both living and dead organisms. The organisms range in size from single-cell microbes such as bacteria and amoeba, to multi-celled nematodes, to earthworms, to plants and larger animals. Did you know that if you add organic matter such as compost to a sandy soil, it will hold on to water and nutrients better? And if you add it to clay, it will drain better. Now, let's take another look at pore space. You can kind of get the clue this is important stuff. And see how they are created with the help of soil microbes. When you have healthy living soil, the particles such as minerals and organic matter, they clump together. And they do this with the help of gluey substances that are exuded from things like bacteria, fungi, and plant roots. These clumps are referred to as soil aggregates. When particles clump together into aggregates, that's what creates the pore spaces. Small aggregates are formed first, then larger ones. Now, when soil is aggregated and has plenty of pore spaces, it is said to have good soil structure, and that's what you want in your soil. So here's a short video that I'm going to show you from the Soil Food Web School that demonstrates this concept.
With the soil food web in place, the picture rapidly changes. Beneficial bacteria produce sticky compounds, or glues, which make it easy for them to adhere to the surfaces of organic matter and sand particles. These particles then adhere to one another, forming microaggregates. Notice how as things clump together, small spaces are created around them. Fungi also produce some glues, and their long, narrow tube-like structures act like ropes, tying together the microaggregates to form macroaggregates. Now even larger spaces are formed in the soil structure.
All right, hope you enjoyed that. Okay, now I'm going to have Kelly and Janet pass around a tray, and it has some jars in it. One of them shows you a soil with absolutely no structure whatsoever, and the other ones all have fairly good structure, some better than others. But you'll see the little clumps, and even one has a space. It's the one around the oak tree, because they're labeled oak tree. It even has a space where earthworms have been crawling through. So that was kind of, it's very dry because it's dry now. I don't water my oak trees, clearly. But you can see how it's still together and has space and roots coming out of it. So enjoy looking at that. So while that's being passed around, we will move on. All right, so now let's talk a little bit about roots, since roots are where the soil and plant interfaces. The area around the roots is known as the rhizosphere, and I just find that's a fun word to say, rhizosphere. Roses have taproots that grow deep into the soil, and they can grow down as deep as 3 feet. And then there are the fibrous roots that grow out more horizontally, and they can spread out about 3 feet. Some of these roots become woody when mature, and these woody roots provide the stability and connect to the finer roots, and they also take up some water. So if you look in that top picture, well, I don't know if you can see it from your seats. I think you can. You can see woody roots and then those little finer white roots. And then down below, the lower picture shows one of those fine roots with a lot of root hairs on it. And the finer roots, they spread out into the soil and they produce those root hairs. And the finer roots with those root hairs are responsible for water and nutrient uptake. So they're super important. And those little root hairs only last for a day or two. They die off and they grow more. And we're going to talk about why that happens in a minute, because there's something very cool scientifically that I will share with you. So when you are adding water to your plants or adding amendments such as compost, the fine roots are, with those root hairs, absorb those nutrients closer to the surface of the soil rather than super deep. And remember, they can be located within 3 feet of your plant. So place your water, your compost, or any amendments within that area. Don't just put it like right next to where the stem comes out of the main stem comes out of the ground. Remember that root system is all the way out. So that's super important. But make sure also that if you put things like compost in, that they're not touching a stem, because it'll rot it. So you need to leave several inches for that. Okay. Another very important job for roots is to send exudates into the soil. Many of these exudates are produced from the process of photosynthesis. In the top photo here, you see it as daytime and the sun is shining. The leaves are taking in the light energy and carbon dioxide and producing sugars. Some of these sugars then leak out of the roots. Those are what are known as root exudates. And you can see these in the lower photo, largely magnified, but it is like little liquid drops of carbon, sugars, all that stuff coming out. And root exudates are important because they feed the soil microbes. And they can also help create soil aggregates, which can cling to the plant roots. So when plants are putting these exudates into a soil that is alive with microbes, the roots will most likely have, another fun word, a rhizo sheath, as you can see in these photos. The rhizo sheath is soil or soil aggregates that cling tightly to the roots. This would not happen without soil microbes interacting with the root exudates. Now, the rhizo sheath is full of beneficial microbial life. It's also got organic matter and minerals. These help the plant withstand drought, and they help keep diseases and pests away. They are also cycling nutrients right there in the root zone, so it makes it easy for the plant. So now let's talk a little bit about those plant nutrients. So plant nutrients are found many places in your soil. They are found in the rocks or minerals. Are you surprised that they are in rocks and minerals? I was when I first learned about it. Many of these nutrients, though, are not available to your plants until soil microbes come along. You can thank bacteria and fungi for making those nutrients available to your plants. But the plants themselves also create some acids that help break down rock material and organic matter. Nutrients are found in organic matter, which consists of dead plants and animals, including those microscopic ones. Think about a dead plant. The plant once contained all the nutrients it needed to live, as well as grow. When it dies and is then decomposed by microbes, those nutrients are returned to the soil. And some of those nutrients go back into living plants. Soluble nutrients can be found in the water in soil, and nutrients such as nitrogen and carbon can also be found just in the air. So let's explore the soil food web and discover how microbes help cycle those plant nutrients. Remember that when the sun shines, plants photosynthesize. Then they produce exudates. And the soil also has dead plant parts, animals, and microbes. The exudates and dead organisms are decomposed and consumed by bacteria and fungi. And these are beneficial bacteria and fungi, not the disease causers. They are there for the purpose of decomposition. The nutrients that were once contained in these materials are now contained in the bodies of the bacteria and fungi. These nutrients will stay in their bodies until they are consumed. Protozoa, such as amoebae and nematodes, are microbes that eat bacteria and fungi. Their bodies take in what they need and excrete the excess into the soil. Now, what's really neat about these excess nutrients that are then pooped out? Well, they are in a form that plants can easily take up. So that's how that system works. There are also very tiny microarthropods, larger arthropods, and even larger critters, such as earthworms, gophers, etc., that contribute to nutrients being cycled through the system. In a healthy living soil, you have a great diversity of organisms. If there is a group missing, then the system does not function as well. For example, if you have a lot of protozoa, but a lot of bacteria, but no protozoa grazing on them, or nematodes, they're not eating them either, then those nutrients don't get released from the bacteria. They just stay there until the bacteria dies. If the bacteria dies, those can be released. When the system is functioning well, though, the plants have nutrients available in small amounts all the time, so when the plant needs them, they are there. Now, this is the cool science part. Well, not that it all isn't, but this is really neat. One more way that plants can get food is through the rhizophagy cycle. And this has been a relatively recent discovery in Australia, but has been looked at more in depth by Dr. James White at Rutgers University. It turns out that some bacteria can actually go inside the plant root where their cell walls are stripped away and the nutrients are absorbed. The wall-less bacteria are then put back into the soil through root hairs. And in the soil, the bacteria reform those cell walls, they accumulate more nutrients, and guess what? They're taken back in the root again. And Dr. White has discovered that plants only form root hairs when this process is happening. And I just found this incredibly fascinating. I imagine you would too. I was like, are you kidding? Anyway, fun thing to talk about. So now let's talk about mycorrhizal fungi for a minute. So please raise your hand if you've heard of mycorrhizal fungi. Okay, about half of you, excellent. Up to 90% of land plants have an association with mycorrhizal fungi, and roses are one of them. Mycorrhizal fungal partners, they partner with plant roots. So you've got the fungi and they form a partnership. The mycorrhizal fungi help the plants get certain nutrients from farther away than the plant roots themselves can get to. And mycorrhizal fungi, and this is important in our drought-stricken California, can also get water and bring it to the plants. So we want to be nice to those mycorrhizal fungi. So if you look at this photo, the plant roots are the whiter and more yellowish color, and the white shows the fungal partner, which is much thinner and can get much farther into the soil where the nutrients are. And why do the fungus do this for the plant? Well, there's a really good reason. Fungi cannot get carbon on their own, and so they need this partnership. So the exudates have the carbon, and then they feed it to the fungi, and the fungi gets the nutrients that the plant needs. Plants with a mycorrhizal partner also tend to be more resilient, meaning that they are better able to fight off disease and they are more resistant to drought conditions. There are many types of mycorrhizal fungi, but roses associate with arbuscular mycorrhizal fungi. And these particular ones actually enter inside a plant root rather than just around it. And they are particularly good at obtaining phosphorus and calcium. And just a note here, when a gardener adds synthetic phosphorus to the soil, the plant takes it up. And then guess what? It no longer needs to feed their mycorrhizal partner. So the relationship between the plant and the fungi is very limited or severed altogether. So now you might like to know if your soil has life in it. So what are some things you can do at home? Well, you can dig up a plant, but I don't recommend digging up your roses. But you may have annual plants or even weeds nearby. Is the soil clinging to it, or is it rather bare? This picture that I have of the rhizo sheath is just a grassy annual weed that comes every year in my garden. I thought, well, I'm going to dig it up and see what happens. And by golly, they were all clinging tightly. And so I went, okay, even under my weedy grass, there's a lot of life there. So remember, that if you see this rhizo sheath, it means it's full of life, full of organic matter, full of minerals. So a lot of good things are happening if you have a rhizo sheath. Then look closely at your garden. Do you have a variety of living creatures, perhaps bees, earthworms, pill bugs, lizards? Well, if you have a variety of living animals, then it's likely that there is a soil food web supporting them. All right, you can also do a slake test. A slake test is when you take a chunk of soil from your garden, dry it out for about a week, then put it in water. I will show you a video of this process shortly. So Kelly's going to pass you around another jar, so I'll tell you about it while she's getting going. It has 2 soil samples in it. They're already quite dried out, and they came from the same places as the soil samples that I'm going to be showing you in this video. One of the samples came from underneath a patio that was just taken out. And the other one was from an area near my house that had butterfly iris, and I never did anything with that area. Just sat there and just was on its own. So as you're looking at them, try to see which one you think was just compacted underneath the patio versus which one was more alive. Well, soil that has been well aggregated because it is full of life will hold together fairly well when you put it in water because of all the glues that were created by the microbes and the plant roots. Soil that is lifeless will just simply fall apart. It will be unstable. So now I'm going to show you that video where I put the soil into water. When it's done, you can tell me which one has life in it. Isn't it? I've just done a house remodel, so I went around looking for dirt clods, and then I went, this should work, and it was brilliant. Anyway, so which one had no life in it? The one that dissolved, just totally fell apart. That's what happens when you do not have life and those aggregates in your soil. All right. Okay, so now that you know that you have living soil, let's talk about ways that a gardener can unintentionally degrade that soil. When soil is degraded, it loses its structure and gets compacted. Notice the photos at the bottom here. On the left is soil that has been untouched and plants just allowed to grow freely. The photo on the right is a neighboring area where the soil was compacted. you can see how little life there is. Just a few weeds that are trying to repair the damage. Yes, that's what weeds' job is. When you've done something not quite right, the weeds come in, but they're fixing things. So how does this happen? This all happens when soil is left bare by excessive digging, too much foot or vehicle traffic, by using biocides, meaning pesticides, herbicides, and fungicides, and using synthetic fertilizers. So let's explore this further. When you leave soil bare, it gets compacted and eroded by raindrops. Yes, raindrops hit very hard, and topsoil can be taken away by wind or water. Water cannot infiltrate, then percolate, and soil gets extremely hot, which is never good for your plant roots. When soil is dug excessively, then pore spaces collapse and fungi are torn apart. All those mycorrhizal networks that you saw, you want to keep them in place. When one continuously walks on garden beds, then pathways are made where plants cannot grow, pore spaces collapse, and water cannot infiltrate. When biocides, meaning herbicides, fungicides, pesticides, etc., are used, they often kill more than that target organism and can create a decline in the diversity of organisms. When the beneficial organisms are inadvertently killed off, it is often the pests that come back more quickly than the beneficials. Using synthetic fertilizers feeds the plant directly rather than nutrients being released by the organisms in the soil. When plants are fed directly, they do not send out those exudates that attract the microbes, so this limits the relationship between plants and their microbial community. And in so doing, natural nutrient cycling is interrupted. And if used in excess, they can leach into groundwater as a pollutant. There are times when you may need or want to use them, and I will address that shortly. In summary, we create degraded or compacted soil when we leave the soil bare without living plants or residues, when we decrease the microbial diversity, and when we dig excessively. So now that you know how the soil ecosystem functions, you know that you have soil life, and you know what practices can degrade your soil, what are some of the best practices for fostering living soil? Well, first is to always keep your soil covered, but do leave little spaces for the native bees. They like bare ground, so don't cover it all. The best way is to have living roots in the soil. To have living roots next to your roses rather than bare ground, you can consider companion plants. And if you have a variety of plant types, this also helps increase the diversity of microbes below the ground. This diversity helps all the plants. If it's not possible to keep the ground covered with living plants, then keep the ground covered with compost and mulch or just mulch. You can put about a ½ inch of compost and cover with 2 to 3 inches of mulch, but keep the compost and mulch, as I said earlier, several inches away from the main plant stem. Limit your digging so you do not tear up your mycorrhizal network and collapse the pore spaces in your soil. Again, a little bit of digging is not a big deal, just not excessively, because sometimes you have to plant little seedlings in there and all. Make sure to walk on pathways, not on your garden beds. If you use a leaf blower, use it only on your hardscape. When you use a blower on soil, you blow away topsoil and you leave the soil bare. This can lead to compaction. It is often a good practice to let leaves stay where they are to feed the soil. But if you have diseased leaves on your roses, you may want to put them in your green bin to be composted commercially instead of leaving them on the ground. Avoid or limit the use of synthetic fertilizers. When you feed these to your plants, the plants will not put out those exudates to attract microbes that will feed them. This is particularly true with phosphorus and mycorrhizal fungi, as I mentioned earlier. Organic fertilizers are fine as they feed the soil microbes, and the microbes provide nutrients to the plants. If you do feel you need to supplement with synthetic fertilizer because your soil is lacking, make sure to use the least amount needed. Your plants may need the boost until the soil food web is working better. And if you encounter a problem in your garden with pests, diseases, or weeds, use IPM, or integrated pest management. Look at the bigger picture about why this is happening and use the least toxic methods first. Use the pesticides, herbicides, fungicides as a very last resort, and remember that they are poisons and will often kill off more than you intend. If you really must use them, then make sure to follow directions carefully. Okay, yes? Oh, yes! Thank you. I'm glad you stopped me. All right, so did you guys figure out, they kind of look similar, but I couldn't, you can sort of see the difference. Did anybody figure out which one was the one that fell apart? This side or that side? I don't know if you can see it from there. Exactly, lighter color. It's very subtle right now. When I first got this out of the ground, it was a little darker, but even if I put in this light, it looks darker. But yeah, it's the lighter colored one. And even this one, you could see, that you could see like potential of little pore spaces in between it. Anyway, thank you, Kelly. Okay, now, I've been looking so forward to showing you this. So right now, I'm going to show you some pictures of roses that are in the ground and in containers. And I, in 2016, bought my first microscope, and I learned how to look at soil microbes and identify general categories. You can't figure out species and all that, but you can see what's happening. You can see if you have life in your soil. And how I do it is I get samples of soil, and then you put about 1 milliliter in here, and then fill water, say, up to the 10 mark. Shake it, shake it, shake it, so you're loosening the microbes off of the organic matter and stuff, and then I put 1 drop under a slide and then look at it. So I'm going to start this going, and I want to thank Karen, who ran around and got me a bunch of soil samples from you guys. So we had fun with it. She even came over and saw my microscope and everything.
I've never seen that.
Yeah, so it was super fun. Okay, so let's go. I'm going to make this bigger. Okay, so first these are in-ground roses, so we'll enjoy that. Okay, I'm going to stop this, pause it for a second. So I just want to show you what you're looking at. So here, these darker pieces here are organic matter, little tiny aggregates. The teeniest, tiniest little dots are most likely bacteria. If we were to look at it a little closely, bacteria are very smooth around the edges. And if they're not smooth around the edges, they're clay particles. So that's just kind of a general sense of what's here.
Is that magnified?
Oh, sorry, yes, this is at 100 times magnification. Oops, sorry, I'm touching the microphone. I shouldn't do that. Yeah, so we'll go on. Okay, and this is for, I guess I put that up pretty fast. This is at 400 times, and this little thing, it wasn't the best picture I've ever taken. This is an amoeba cyst. So this would be an amoeba, but it has gone dormant. It could have been just from shaking that it went, oh no. But anyway, so in this rose, we know there's bacteria. We know there is at least some amoeba. Let's keep going. Okay, so, oh no, it went back to the beginning. Sorry about that. We'll just let it run. Okay, then here is another kind of amoeba. It's an Arcella. It actually looks like it may not be that healthy. But anyway, this is what's called a testate amoeba, which means it has a shell, like a snail has a shell, and the amoeba lives inside. And this is the little hole where it can come out and feed, and then it goes back inside. And this here, if I could focus it a little more, is another kind of bacteria called an actinobacteria. And that looks like another little aggregate that's formed. Oh, and this was so cool. I actually got a video, but I didn't want to scare you. This is a little mite, and this is the end of the slide, and there was a little water that got out from under the slide, and this little mite was there crawling around and moving around. It was crazy, but they often will eat fungi. So they're part of the soil food web. Yeah, it was fun. Okay, so another rose in the ground. And okay, so this one, so you can see, nice big aggregate there. Well, it's still probably a micro aggregate because this is really blown up. But, and then this is a piece of fungal hyphae. They look just like sticks, and you'll see more as it comes up. And again, you know, lots of bacteria and stuff in there. And let me get it going again. Okay. Without me touching, do you spot two fungal hyphae? Yeah, two pieces of fungi, top and bottom. Okay. So these roses already, you can see there is life in the soil. And this is a nematode that, yeah, so it would most likely eat fungi. So now we're going to look at roses in containers. A beneficial one, yes. Although that one's tricky, but I won't go into that. Okay, so I love this. This whole thing is a beautiful fungal hypha. These are called septate or septa, so it's septated. And from what I understand, this is like a higher order fungus. And anyway, I just think they're just beautiful. And there's another aggregate with all sorts of fungus sticking out of it. And then this Arcella I thought was amazing. So that's why I said the other one I think was kind of not maybe alive. But this one is definitely alive and just gorgeous. And then this is another type of testate amoeba right here. This is its opening. And a lot of times I see this kind in Arcella sometimes. It could be a fungal hypha, but it's hard to know without focusing in on it. Sometimes the plant material also looks like fungi unless you focus in on it. Okay, and then here's another one in a pot. Okay, so here, do you see the fungi? You guys are getting good at microbes, right? And then you see the amoeba. And then here, I can't tell you exactly, but it looks like a fungal spore, most likely. Don't know exactly what kind, but you do see fungal spores. And another testate amoeba. And another one, oh, this, I wish I could have gotten a better picture. These kind have the most beautiful outside to them. And another Arcella, so another amoeba. So this, okay. So I hope you could see that these were just roses from your guys' gardens and such life in them, in containers in the ground. There's life, and that's what you want to see, and they will support themselves. Now we're going to look at a rose that has been neglected for a year or so. It has had no mulch on it. The soil is bare. The blowers have been used on it. So let's see what it looks like. I'm not telling where any of these came from. So you can see it's a little stressed. Okay, we're going to stop here. Now you're going to look here. Do you see those dark brown aggregates anywhere? No, you really don't. This is at 100x. Bacteria, yeah, because bacteria show up no matter what. Well, gosh, if there's no bacteria, you're probably at Chernobyl or something. But there's always bacteria. But yeah, but there's nothing much here but minerals. So yeah, I didn't point out minerals before. But the things that kind of reflect, yeah, are what am I trying to say? Refracting the light. Those are minerals, the sand, silts, and clays, right? Well, clays are tiny bit. Okay, and then here we're going to go up to 400x. What are you seeing? Almost nothing but what look like rocks because we're magnifying so much. So these are mostly minerals. Okay, maybe there's a little organic matter, possibly, but I think if I focus on them, I'm going to find that those are more probably sand particles. So this is a compacted soil, okay? Because remember, this has been shaken and so it's spread out a little bit, but even though it's been spread out a little bit, you can see, and I looked through the entire slide, found no life, no fungus, no amoeba, no nematodes. So yeah, so it was really, yeah, it really kind of told a story. So I'm glad you enjoyed the video because, as I said, it was really fun just seeing what was there and all the different roses, but also what a difference your management makes to the underground world. Okay, so let's do a quick recap of what we covered this evening. Remember, when you foster living soil to grow healthy roses, your soil will have a diverse soil food web, good soil structure with lots of pore space. Water will infiltrate and percolate, and water will be held in soil for later use by the plants. Nutrient cycling will happen with the help of living plant roots and microbes. And now that you know about living soil and how to care for it well, I hope you enjoy caring for your soil as much as you like caring for your roses. All right. So you will be getting, like I said, a PDF of this later on, but here are just some references, including the second one, which is the soil texture field test. And you're going to look at it, let's say, the sheet like that, like I showed you before. It's super fun to do if you've never done it. And I didn't put it on here, but you can also do something called a jar test. So if you want to look up jar test, you can do that. And again, thank you to the Peninsula Rose Society for hosting us this evening. It's been so much fun. And thank you, Janet and Kelly, for helping out. And I want to thank Kelly Troikai, Terry Lingzo, Karen Flores, and Stu Dalton, who all kind of went through my presentation and gave me feedback and all that, so it helped make it better. And again, Karen talked about all this, but you can stay connected and consider donating if you would like. And if you have a question, ask a Master Gardener. We have helpline on 3 different days, and the information is here. And that is it.